Vamos a iniciar. En nuestra serie de seminarios institucionales, el día de hoy tenemos el placer, o tengo particularmente el placer de presentar a la doctora Jolene Ramsey, la cual es profesora asistente en el Departamento de Biología, de Ciencias Biológicas, perdón, en la Universidad A&M de Texas, donde se incorporó en el 2022. La doctora Ramsey es microbióloga y viróloga especializada en bacteriófagos y su investigación se enfoca en comprender cómo estos virus infectan y provocan la lisis en las bacterias que hospedan. Obtuvo su licenciatura en Biología y Español por la Universidad Western de Colorado y su doctorado en Microbiología por la Universidad de Indiana Bloomington y realizó su investigación postdoctoral en el Texas A&M University. Su trabajo combina virología molecular, genética y genómica para estudiar las interacciones fago-hospedero y el mecanismo de lisis con posibles aplicaciones en el control de bacterias resistentes a antibióticos. La doctora Ramsey tiene una larga lista de publicaciones donde podemos encontrar revistas como Journal of Virology and Bio, Journal of Bacteriology, Microbiology and Molecular Biology Reviews, and phage. También es una de nuestras socias más participativas de la Sociedad Mexicana de Virología. Así que, pues para mí, como ya les he mencionado, es un placer el tenerla en nuestra serie de seminarios. Al final tendremos un pequeño espacio para que puedan hacer las preguntas a través de nuestros micrófonos. Muchas gracias, doctora.
Bueno, muchas gracias, Carlos, por la introducción y a todos por tenerme aquí. Voy a presentar mi seminario en inglés, pero al final, con gusto, respondo preguntas que vienen en español. So today I'm going to tell you about bacteria and bacteriophages. And to start, I would just like to say that bacteria are everywhere in the planet, and that is really important because that includes everything from the bottom of the sea to the top of every mountain, soil, plants, animals, and including the production of food that we eat. Now, that's not all that they do in the environment. They do cause us many problems, including things like I have shown here, and that's simply just living in their niche, like when they form biofilms on our teeth or when they form biofilms in the built environment, like this picture here where I'm showing in the International Space Station, there are biofilms that are clogging the water recycling system. And that's one kind of problem that bacteria can cause us, but they also cause infections in the plants that we eat, such as the bacterial infection of this wheat plant here. And more close to home, they cause infections in ourselves and in our loved ones. Fortunately, they're not the only ones out there, and they are outnumbered in our universe by bacteriophages. Now, we're showing here estimates of the total number of bacteria and bacteriophages on the planet. So phage are about 1 to 10 outnumbering bacteria, and phage are, of course, viruses that infect and kill bacteria very specifically. They come in many different shapes and sizes, as I'm depicting here, but they have different kinds of genomes, just like eukaryotic viruses, DNA, RNA, large, small, and they can also infect and immediately kill, or infect and integrate and not kill immediately. So ever since we have discovered these viruses over a century ago, scientists and physicians have been asking if the enemy of our enemy can be our friend, and can we tip the natural balance between phage and bacteria in the environment in favor of the natural phage predator. So I'm going to show you some examples where we have been able to successfully do that. This first one here are several products produced in the food safety industry. So we have bacteriophages that can target listeria, and these listeria frequently cause bacterial infections on fruits like cantaloupe. We also have bacteriophages that target E. coli. E. coli are frequently a problem on cut meats. And then third, I'm showing here phage cocktails that target Salmonella, and Salmonella are frequent causes of foodborne illness, not just in humans, but also in pets. And so here I'm showing that these can be used in prevention of Salmonella contamination of pet food production. Another example is in our food production, such as in aquaculture. It turns out that when you grow or farm shrimp or fish or other kinds of creatures of water, then they can become infected and those infections spread very quickly, and that can lead to great losses, both of the animals as well as to the profits. And so bacteriophages have been produced to target various different vibrios that cause infections in shrimp, and that has helped to salvage the product that they are producing. Additionally, I would like to give you this example in plants. So there are bacterial infections of plants. The example here is Xylella fastidiosa infections of grapevines that are used to produce grapes for wine. And these xylella are very difficult to grow, but in this case here, this was more close to where I am. The Center for Phage Technology isolated bacteriophages and produced a device for injecting these directly into the grapevines to prevent the growth of the bacteria and allow the grapevines to produce grapes. Interestingly, I'm not sure if this is a success yet, but the beauty industry has started to take interest in bacteriophages as potential applications for preventing acne. So you can see here two products that are produced and distributed through Sephora. These are meant to be to be applied on your skin and to prevent the growth of Propionibacterium acnes, which causes acne. And so I'd really love for the Sephora and other producers of these products to become more interested in some of the things that we work on, because I imagine that these will be relatively lucrative. And now to turn kind of where I'm more focused in my lab, and that is we have had successes in using bacteriophages to treat bacterial infections, in particular cases where these bacteria are resistant to the antibiotics that are normally used in the clinic. So I'm showing here the example of Dr. Tom Patterson. He was a professor at the University of California, San Diego. He's also pictured here with his wife, Dr. Stephanie Strathy, who's also a professor. When they were on vacation back in 2016, he contracted an infection that was resistant to all available antibiotics. By the time he is shown pictured here, it was an Acinetobacter baumannii infection. And through a series of emergency consultations with the physician, who's also shown here, his hands are shown, Dr. Chip Schooley, and scientists here at the Center for Phage Technology and in the Navy and at the and another university in San Diego, they were able to find and produce bacteriophages that ultimately did help him to wake up and come back to a state of health. This story is told in the medical thriller that Stephanie co-authored with her husband called The Perfect Predator. I highly recommend it as an excellent read to get an idea of kind of what was the state of affairs at that time. At that time, when she found a way to make this treatment available for her husband here in the United States and in most Western countries, phage were not regularly used or even known of as options for treating bacterial infections because of antibiotics. Back when the world wars were happening and we discovered antibiotics and they began to be produced, phage were kind of left by the wayside in terms of development for use in clinical medicine. But there were still countries who have been using it ever since the early 1920s to try to use them for treating bacterial infections, and there have been successes in doing so, but they haven't become widespread enough for most of us to be able to access in the clinic. It is important to consider how we can develop them, though, because these antibacterial resistant infections are becoming more common, and the list of bacteria that are a problem in this area is growing. So I'm showing here a couple of lists that come from the Centers for Disease Control and Prevention, but these are very similar to the lists that are produced by the World Health Organization. You can see that there are listed primarily bacteria, so Acinetobacter, Clostridioides, Enterobacteriaceae, Neisseria, and all of the ones listed over here. There are a few fungi listed, but most of them are bacteria, and they're primarily gram-negative bacteria. These are listed urgent and serious because they are either resistant to most or almost all of the antibiotics we have produced and ready to use in the clinic. And they are causing quite a large number of deaths or associated with deaths that are found in all areas of the globe. So this is dividing up deaths per 100,000 by specific kinds of antibiotic-resistant bacteria. And I've pointed out my current home here in high-income North America next to several other Central Latin American countries. You can see that even no matter where you go across the world, everyone is having a problem with this. And some of us are more affected than others, but we are all heavily affected. So pursuing alternative options for either prolonging the life of antibiotics or using in place of them is a very critical thing to do. Now, I mentioned that in other countries, they have been using bacteriophages for a long time. A few years ago, I had the privilege of traveling to Tbilisi, Georgia, to celebrate their 100 years of a bacteriophage therapy clinic. And when you walk into their pharmacy, you see this, which is a refrigerator full of human therapy grade ready bacteriophages for treating anything that you want. These are sold over the counter, meaning you can go in and buy them without needing a prescription. And so they certainly have been using them for a long time. And the question is, what is the difference between being able to walk into a pharmacy in Georgia and buy these off the shelf and the case here? Well, there are quite a few things that make us have to slow down in terms of thinking about how we can use bacteriophages for therapy in the clinic. So I would call this a tope or some speed bumps, like the armadillo that is the Texas speed bump here. I'm not going to go through these in great detail, but I just want to mention that there are many challenges to us being able to regularly access phage therapy as an ancillary or primary treatment in the clinic. And so some of these challenges include the time. So the time from when a person knows that they need phage to when they can get it can be from months to years, which is not good when it's an emergency situation. Bacteriophages, just like antibiotics, are frequently going to pop up with resistance in their bacterial hosts. And in fact, phages become unable to infect bacteria even faster than antibiotics. So we have to consider combinations and other ways to get around that problem. Numbers, we don't have very many good comparable cases. There are many individual case reports, but having high numbers of clinical trials that we can use to determine treatments are lacking. It's difficult to know how to produce them because they're all very different. Bacteriophages are very specific to their host, and therefore you have to develop a new pipeline for producing each of them. I would guess that many of you prior to today didn't really think very much about bacteriophages as an option for treating bacteria or bacterial infections. And so there is an awareness problem that makes this a difficult thing to deploy. And even more importantly, there's an access problem. Not everyone is going to be able to access these therapies. They're expensive. And part of the reason they're expensive is because we can't mass produce them, and there's no way for companies to make money, and therefore there are not very many that are pursuing that in terms of human cases. Every government has a different way of approaching their approval of these kinds of treatments, and that is something that takes a long time to develop and to be able to get approval on an individual basis. And then there's a lack of research in many different areas, and so I'll give you an idea of some of those. When this does happen, it involves a relationship between a patient and a physician. And that physician has to first determine what is the pathogen of interest that is infecting this individual before they can even go forward with looking for a bacteriophage that can specifically kill it. And then they have to partner with either researchers or companies who are willing to find the effective phage from the environment or from the collections that they have in their personal laboratories. Those then have to be screened for the safest type. Not every phage is going to immediately infect and kill the cell. Therefore, you need to be able to choose the correct kind. Sometimes we have options that can infect a bacterium, but that are not immediately going to kill, and we can modify those genetically to make them suitable for this purpose, if that is allowed in the country where the patient is located. We can also apply antibacterial proteins from those kinds of bacteria that are infecting the patient without having to use the phage themselves. And then once those initial steps have been gone through, we have a phage or multiple phages that are useful for this particular patient. You have to make enough, and it has to be pure enough to use in that patient, which not every lab is set up to be able to do. And then we need to get approval for human use. And this requires getting those appropriate paperworks filed with the government and with the hospitals that are involved in the treatment and care of that patient. And then finally, we have to know how much to give each patient, how much, how often, where do we give it, and what do we do when the inevitable resistance begins to arise. So all of these are areas that the field is working in. And I'm going to focus today on the areas where my lab is primarily doing research, and these are in finding phage, choosing the right kind of phage, applying proteins that kill, and then how do we make enough to be able to use. So this is kind of the way that I divide up the research in my lab. We do a lot of discovery and genomics, but I'm not going to talk very much about that today. I'm going to focus on my area of research in bacteriophage lysis. So this is how do they specifically kill bacteria? This is what makes phage therapy and other applications of phage even possible. So what are the mechanisms that they're using to kill those cells? And I'll tell you a couple of different stories. But before I get into those, I'll give you the specific background on how lysis in bacteriophages work. So this is a growing flask of E. coli in the lab, and I'm monitoring the optical density or the turbidity over time. You can see that these cells are growing, and at a very predictable time, based on the phage that is infecting this culture, you will see by eye that the flask goes completely clear and the optical density drops down to below detectable. And the reason that this is genetically programmed and so rapid and synchronized is because there are specific proteins that are orchestrated to trigger at the time when it's ready for the phage to leave the cell. So to zoom in a little bit further on this remarkable process that you can actually see by eye, it still looks really cool when you look at it at an individual cell level. So these two different E. coli cells have been infected. They're at this stage where they have phage inside. They just haven't lysed yet. The one on the left is sped up. And you can see that this one is infected. It's going to slightly change shape, and then it will burst to release the phage into the environment. And this one on the right is actually slowed down. Here you can see that the cell did not lose its shape. It simply burst from the end and is releasing all of the interior contents of the cell, the phage and everything else, and it's simply crumpling up. And you can see here, not even 3 seconds have passed. So when this does occur on an individual cell basis, it's very rapid. And although the outcome is the same in both of these cases, the phage have escaped the cell, the specific way that the phage compromises the cell to get out is different. And that's really what we're interested in, is what is the diversity of ways that phage are able to escape from the cell. And I'll make the important point that these cells did not burst because they are completely full of phage and the pressure causes them to pop open. There are active mechanisms that the phage are using to escape through the cellular barriers. And in my case, studying gram-negative bacteria, there are 3 layers of that cell envelope. Those 3 layers are the inner membrane, which is an energetically charged membrane, and then there is this periplasmic space that contains a rigid layer called peptidoglycan. This is a cross-linked meshwork that gives the cell its shape and resists a significant portion of the turgor pressure that is normally inside of a cell. The peptidoglycan is usually linked either covalently or non-covalently to the inner membrane as well as the outer membrane, and they work together to help the cell to maintain and resist the pressures from the inside and the outside. Gram-negative cells in particular have this outer membrane barrier. This is another lipid barrier that is an asymmetric lipid barrier. It forms a permeability barrier between the cell and the exterior. This is the reason that most gram-negative bacteria have a higher intrinsic resistance to any antibiotic that we would like to use. So phage have evolved with their gram-negative hosts for a very long time, and they have specific mechanisms to target each of these layers of the gram-negative cell envelope. They have membrane proteins, soluble enzymes, and then proteins that can actually span this gap. This blue one is called an endolysin. It's an enzyme. This green one is called a holin, and this white one is one of the spannin proteins. I'll tell you how these work. So the holin is the one that really controls the timing of lysis. It is produced into the inner membrane, and it accumulates there harmlessly until it reaches a certain critical threshold concentration. And when that occurs, it will rearrange and form very large holes. These holes are then going to mix the interior and the periplasmic space of the cell and effectively kill the cell. The cell is dead, it cannot make energy, but the phage are still trapped inside. So when this hole gets made, the endolysin is released into the periplasm where it can access its target, which is the peptidoglycan. It begins to enzymatically degrade that layer, and that's why sometimes you see that the cell starts to lose its shape because it is no longer held together by the peptidoglycan meshwork. And then finally, there is an outer membrane barrier, and that also must be actively disrupted before the phage can escape by spannin proteins in this case. And these work very similarly to eukaryotic fusion proteins, such as the ones that viruses use to fuse between a host cell envelope and the virus envelope, or such as the ones that some eukaryotic cells use for fusing vesicles. What they do is, because they're embedded in both membranes, is after this peptidoglycan meshwork is removed, they undergo a favorable conformational rearrangement to pull the two membranes together and ultimately fuse them, which causes a local position where all of the turgor pressure from the cell can escape, and that is what releases the phage. Now, the very different kinds of phages are actually united in that almost all double-stranded DNA and double-stranded RNA phages that we know of use this pathway that we call multi-chain lysis, where they have some sort of hole, and that can be a large hole like the one that I showed you, or it can be a very small hole. They will encode an enzyme such as the endolysin that I showed you, or this other form of endolysin that can be tethered, and those will degrade the cell wall. And then we have found that there are not just the spannins that can disrupt the outer membrane, but I'll tell you today about some cationic peptides that can disrupt the outer membrane. And then there are regulatory molecules, and I'll tell you about one of those today as well. And this is somewhat in contrast to the single-stranded DNA and single-stranded RNA phages that use a single gene to inhibit peptidoglycan biosynthesis. And in growing cells, that results in the immediate loss of cell integrity when a cell tries to divide and can't build its peptidoglycan layer. So to understand how we might be able to take advantage of these proteins that phages have made that can already kill bacteria or target a specific layer of the cell envelope, the projects in my lab focus on different phage that exhibit these characteristics. So the first project that I'm going to tell you about today is the disruptin project. In this project, we're focusing on how does the phage disrupt the outer envelope of the gram-negative cell. And I mentioned before that this is an essential step. So to try to convince you of that, I'll show you these data here. So this is phage lysis, like I showed you before. There is an immediate drop in optical density at the population level. And at the single cell level, individual cells are going to go from rod shape to crumpled up rods. Now, when you have an infection with a phage that cannot make spannins, but it can still kill the cell and it can degrade the peptidoglycan, instead of seeing a complete drop in lysis at the population level, you just see a slight drop. And individual cells actually die due to holin activity in its inner membrane, and then they lose their shape due to endolysin activity, but they do not completely burst open. These cells are bounded by an outer membrane, and they cannot release the phage that are trapped inside the cell. So every phage needs a way to get across the outer membrane actively. However, not all phages, when you look in their genomes, so we looked here at a set of almost 700 phage, have spannin genes in them. Most of them do, but a significant fraction do not. And the question that we had was, how else do phage disrupt the outer membrane? And could we take advantage of that to make this barrier that prevents antibiotics from getting in more vulnerable to treatment? So this was something that was started by an undergraduate, Ashley Holt, and one of my co-conspirators as a postdoc, Jesse Cahill. And what they did was they found this phage, an E. coli phage called phages. It was isolated from horses, and it encoded a holin gene and an endolysin gene, but no spannins. What they saw was that when they infected its native host with the phage, they got lysis synchronously, rapidly. The cells went from rods to crumpled up rods. And if you actually watch lysis, they lysed quite rapidly as expected. So that suggested to us that it did have an active mechanism for outer membrane disruption, and we wanted to know how it was doing it. We took our clue from that little gene that's between the endolysin and holin, number 28. And I will show you here an assay that we used to determine if that was the gene responsible for outer membrane disruption. So we used a phage that was encoding a holin and endolysin, but its spannin genes had been genetically inactivated. And in this case, when you induce this phage for infection, what you normally see when you have no complementation is that the cells become spheres and the phage are trapped inside. If you then add back these missing genes on a plasmid, you get outer membrane disruption and lysis, and this allows us to screen for any gene that we think might be able to disrupt the outer membrane specifically and look for lysis. So we put the phyket GP28 gene in this assay, and we were able to see that when the GP28 gene was present, this red line here, that it behaved exactly like the complementation of the native spannin genes. The cells lysed completely. But if we put in an inactive copy of the 28 gene, we got spherical cells, suggesting to us that it was specifically functioning in outer membrane disruption and allowing for lysis. So our model was that in phyket, there is the holin, the endolysin, and the disruptin. And the disruptin is going to get made just like the other proteins in the cytoplasm of the cell. And then that protein maybe is getting translocated to the outer membrane during the lysis process, and that might allow it to weaken the outer membrane and allow for lysis to occur. So I'm gonna give you our path here, which was an interesting one. So what we noticed was that this protein, I'm showing the entire sequence here on the top, it's 56 amino acids long. It was highly cationic. It has a +8 charge overall. And our collaborators have shown that it adopts a primarily alpha helical conformation, partially embedded in the membrane. We noticed that this was quite similar in properties, not sequence, to a human peptide called LL37. LL37 is a cationic antimicrobial peptide produced by our innate immune system and secreted to protect us against bacterial and enveloped pathogens. And so we know that LL37 disrupts membranes, and we wondered how similar actually GP28 might be to LL37. I'm just going to show you a couple of assays that we have used to look at the function of GP28 and how it might be like LL37. One thing that is commonly done to look at this is mixing peptide directly with bacteria, and then allowing the bacteria to grow for up to 16 hours, and then simply measuring growth by optical density as well as viability by plating them for colony forming units. So we did this assay with synthetic LL37 and synthetic GP28. And we looked to see if it would simply be capable of either preventing growth or even killing, like LL37 does. And what we saw is that at equivalent concentrations in the E. coli host that GP28 is normally working in, we got equivalent amount of killing. When we added 16 micrograms per mil of GP28 or LL37, the bacteria could no longer grow. And this concentration between 8 and 16 micrograms per mil is actually what has been measured for LL37 concentrations in human lungs during infections. So this suggested to us that the GP28 peptide by itself could compromise bacteria sufficiently to prevent them from growing, and in another assay I'm not showing, even to kill them. One of my graduate students, Michael Deborah, has extended this beyond. Here I'm showing a panel of E. coli. So this one down here, E. coli 4S, is the one I just showed you before, and we're comparing now to another cationic antimicrobial peptide, polymyxin B. So here you can see GP28 can kill at 16 micrograms per mil for this particular E. coli. And you can see that it also does that for a variety of other lab E. coli strains, as well as Klebsiella, Pseudomonas, Salmonella, and not for Proteus mirabilis. So these are gram-negative pathogens that are being killed by external concentrations between 16, 32, and 64 micrograms per mil of GP28 peptide. We also tested a panel of uropathogenic E. coli clinical isolates from our collaborators as well as the lab strain. And we saw a similar result where GP28 is able to both kill and inhibit the growth of these organisms, suggesting to us that GP28 is a broadly active antimicrobial peptide. In data that I'm not showing you, GP28 is not toxic against eukaryotic red blood cells, suggesting to us that we might be able to pursue this as a potential antimicrobial peptide on its own. So we've been pursuing understanding how GP28 works at the molecular level, and I'm not going to tell you any of the experiments there, but I'll show you what our model is. And the way that we are thinking about it is that GP28 is made in a cell that has a very good inner membrane that can resist the inner turgor pressure from the cytoplasm, partly due to its connections to the other two layers of the cell envelope. And the very outermost layer of the cell envelope, the outer membrane and the outer leaflet, has lipopolysaccharide, which is a specialized lipid that I've drawn in gray over here. And it includes not only the lipid component, but a complex carbohydrate. And that carbohydrate is modified by phosphates that give it a negative charge. These negatively charged phosphates are cross-bridged by divalent cations such as magnesium. And because of that extensive cross-linking in the outer membrane, that gives it a very good permeability barrier. So the way we're thinking that GP28 might be working is it's either directly interacting with LPS, since it's highly cationic. We hypothesize it is displacing the divalent cations that normally cross-link LPS, and that would disrupt this LPS barrier. And that would allow the turgor pressure that is released from peptidoglycan and intermembrane being removed by the other phage lysis proteins, allow that to cause lysis during a phage infection. But we've also seen that GP28 works by itself. So we're hypothesizing that GP28 not only works at the outer membrane, but that it also has an inner membrane target that permeabilizes that. We've seen that just GP28 treatment allows for the entry of dyes from the outside all the way to the inside that can stain DNA. So I'm happy to talk more about this story later, but what I'm going to do now is shift gears to tell you about another story where we've been interested in how phage control their own levels of production at the level of lysis. And this is of interest to us because if we're going to produce phages for use in applications, we need to know how to produce a lot and do it cheaply. So I've told you that when a phage infects, it converts its host into a factory. And in usually less than an hour, it'll produce about 100 virions. There is certainly a range, but this is a pretty typical number that we see in the lab. Now, when this lysis occurs, then no more phage get made. Those phage go on to infect other hosts. But phage can also regulate this step of lysis initiation by detecting extracellular signals. One of those extracellular signals that they detect is host cell density. When host cell density is at a particular level, then phage can intentionally delay their lysis and instead increase the production of phage inside of that living cell until favorable conditions emerge. And when this delayed lysis occurs after an hour, and it can increase from hundreds to thousands of virions instead of 100 virions per infected cell. This would be a major advantage for us if we can simply find out how phage are causing at least a 10-fold or more increase in production without changing anything else. What's the ecological advantage for the phage in doing this? Well, when a phage chooses to lyse its cell, it can go on to have exponential amplification by finding new hosts, and every new cell will produce 100 new phage. However, not every environment has new hosts available. And so by staying, they can maximize the use of the resources in the currently infected cell at the risk of potentially missing fresh hosts that are available in the environment. So one case where this has been studied is called specifically lysis inhibition, and it is known that the indirect signal for host density is how often a second phage of the same type infects an already infected cell. We call this superinfection, and when a superinfection occurs, the first phage can interpret that signal as low availability for phage in the environment to infect new hosts. So how do they detect that, and what do they do to inhibit their own lysis process is what I'm studying in this project with another E. coli phage called N4. So N4 does lysis inhibition in the best way. What you're seeing here is another lysis curve. So here are uninfected cells. This purple window is the time when phage will usually infect and lyse the host cell. But instead of lysis during that time, you see continued growth, and then you see a leveling off. And after a few hours of infection, you see a very asynchronous lysis of the population. If you look at the cells at this 180-minute mark, the cells themselves are larger. So this is a formerly acquired cross-section TEM. And you can see that inside of these cells, there are tons and tons of phage that have accumulated. And you can also see when we quantify the actual sizes of these cells, these lighter gray down here are uninfected cells, and the darker gray are infected cells. They're both wider and longer, and they produce up to 3,000 phage per infected cell before they lyse and release those. So we want to know, how is N4 doing this lysis inhibition process? And I'll show you just one more lysis video to convince you that it is actually doing lysis in an active fashion. So you can see that there was a slight shape change right before the cell burst open to release all of the phage. When we initially started this project, nothing was known about the way that N4 did lysis, although it was known that there was an endolysin gene and that there might be some spannins in the genome. So what we found when we looked next door to those genes in the genome were two additional genes that looked like they might be the holins, which we thought were important because we knew that inhibiting a holin could inhibit the lysis process. So to determine whether these genes were functional for lysis, an undergraduate that worked with me, Cody, cloned them onto a plasmid and then put that plasmid into E. coli and allowed for lysis to occur. If they were competent for lysis, we would expect lysis. And that is actually what we saw. So cells without the plasmid, or that are not infected with a rapid lysing phage like N4, just grow, but lysis cassette addition caused rapid and complete lysis in an hour, which is not what we see in the phage. And then what was even cooler was when we infected these cells with N4, we could then inhibit the lysis process, telling us that the phage was producing or some sort of inhibitor, and that the genes made on this lysis cassette were detecting that inhibitor of lysis. In experiments I'm not going to show you, we did confirm the individual functions of each one of the genes in the lysis cassette, and we can tell you that this GP63 gene is a pinholin, the GP62 gene is a holin. So N4 is encoding two different holins, which might have different functions during its lysis and lysis inhibition pathways, and then the endolysin and the spannins. We know that during rapid lysis, this GP63 can form a hole, and that releases the endolysin to degrade the peptidoglycan, and then we get rapid lysis by action of spannins. What we want to know is, how does lysis inhibition happen? And fortunately for us, there was a mutant that allowed for rapid lysis to occur. And we acquired these mutants, but they had been passaged since the 1960s, and there were multiple mutations in the genome. So it wasn't entirely clear what the difference was, but we had seen that the difference between the wild type that does lysis inhibition and the mutant was a change in plaque size. So here in this graph, I'm showing you that the wild type that inhibits lysis has smaller plaques compared to the ones that do lysis very rapidly, which might be counterintuitive, but the way we interpret this is that the rapid lysis allows for quicker spread through an agar plate. And I failed to explain that. These are bacterial lawns that have grown here everywhere where you see it being opaque. And then in the clear spots, this is where the phage have infected and killed the cells. And that allows us to determine how many are present. So taking advantage of this phenotype, a series of undergraduates and other students and technicians have worked on this project looking for changes in plaque morphology. We just very simply infected E. coli with N4 and then collected the phage after a very short time of infection. We did not do any mutagenesis, and we just selected for changes in plaque morphology, isolated those, verified that they individually had rapid lysis phenotypes, and then sequenced them to figure out what had changed. This is the profile of changes that we detected. I'm going to primarily focus today on the lysis genes and these silent mutations. And if anyone's interested, I can talk later about the ones at the terminal repeats. So the first class of mutants that we found were changes in the endolysin. So this endolysin is tethered in the membrane by something called a SAR domain. This SAR domain is weakly tethered in the membrane and comes out when the Holin triggers. In our mutants, we had negative charge in the middle of the membrane domain, which we think would cause it to no longer stay anchored in the membrane. It would come out and degrade the peptidoglycan much quicker than it does during a wild type infection. The second class of mutants were found in the pinholin. The pinholin is a single pass transmembrane protein, and we do not know how many subunits are involved in its oligomerization to form a whole, but all of the mutations that we found were clustered in a single region. So the transmembrane region should be in the part that is colored in blue, and all of the mutations were in this flexible loop here that we think is in the cytoplasmic region. So we are hypothesizing that these are involved in the detection of the signal that is delivered by a superinfecting phage, and that will cause inhibition of lysis. Most of these are serines and threonines, and we're pursuing a hypothesis that involves a phosphorylation cascade. Now, this last class is where I'll spend the rest of my time. We initially thought these were silent mutations in the large virion RNA polymerase that is encoded here. This is the characteristic gene for the N4 bacteriophages, but that idea changed when my graduate student, also named Michael, did an RNA-seq experiment. He took time points over the course of an infection and then simply mapped those RNA reads onto the genome. So what you're seeing here are positive sense mapped and negative sense mapped reads across the entire genome. In an uninfected host, we do have a few reads that are similar to N4 genome. And then the rest of these are N4 wild type and N4 rapid lyser with the reads mapped across the genome. So you can see in the map that on the left side, most of the genes point to the right. And on the right side, most of the genes point to the left. So we'd expected to see red on the left and blue on the right. And we saw that. But we also saw these really interesting red peaks here. So there were antisense transcripts mapping onto the right side of the genome. And those happened to be really close to where we had seen all those silent mutations. So when we took a closer look, it turned out that there were two open reading frames, antisense, to the very large RNA polymerase gene that is essential and that is always made. And these both happen to map directly over the peaks where we saw transcripts coming from the antisense strand, and all of the silent mutations that we had found in this virion RNA polymerase GP50 were in fact encoding nonsense mutants in this open reading frame we have now designated GP75. When I say they were nonsense mutants, I'm showing you here in this table that in GP50, for example, this was a silent mutation, but on the opposite strand for GP75, this codon became a TAG and therefore encoded for a stop. And you can see that these stops were distributed throughout the protein. And so we tested whether or not complementing the phage that were missing this GP75 would restore the lysis phenotype. So we had phage that were mutations with mutations in the GP75 gene, and we simply added GP75 back on a plasmid. What we saw was that in a rapid lyser infection, you get rapid lysis, but if you add GP75 on the plasmid, you can get inhibition of lysis, suggesting that this did complement the phenotype that we expected. Now, these were not annotated in any genome because they're on the antisense strand, and most of the time you don't look for genes that are encoded in the opposite direction where a gene already is. So I had an undergraduate VED who took this on as a summer project when he was supposed to start with me in pipette, but he hurt his arm, so he couldn't pipette. So instead, he sat at his computer and manually looked through about 100 different N4-like phage genomes and found those and aligned the GP75-like genes. And this is the summary of that alignment. You can see that then in pink is the conservation plot for those 100, and the sequence on the top is the N4GP75 sequence. Surprisingly to us, the proteins were pretty highly conserved across those 100 phage that infect a variety of different bacteria. And they all seem to have a conserved C-terminal anchor at the end, according to this hydrophobicity colored plot. And that was consistent with a prediction of a transmembrane domain from Phobius, as well as a Western blot that we are showing here, where this GP75 that is his tagged is produced, and we can detect it, and we see it in the membrane fraction. When we model this, however, by AlphaFold, we get a really crummy score, and that's why it's all yellow and orange. It just models an alpha helix that corresponds to the C-terminal anchor and potentially a folded globular domain that we suspect will be in the cytoplasm. So what we wanted to know was, does this GP75 protein directly inhibit either of the holins of N4? So the little bit of evidence that we have in that direction was collected by another undergraduate, Matthew. So he took advantage of the lysis cassette plasmid system we have and simply added the GP75 gene on either the C or N terminus of this cassette and asked, does it do lysis or lysis inhibition? What do you think will happen? Okay, this is what he saw. So in the case where we have the complete lysis cassette, we get rapid and complete lysis. And when GP75 is added to the N terminus, he saw lysis, but it was later. It was delayed by 20 minutes, which is significant in lysis time. That's almost a 50% increase more. And then we got rapid lysis. When he had GP75 at the C terminus of the lysis cassette, we saw lysis initiate at the same delayed time, but the actual dynamics of lysis were quite different. And so we're in the process of determining whether this might have something to do with the levels of GP75 protein produced in the C versus N terminus construct, or what else might be able to explain the fact that we get some lysis inhibition, but a difference in dynamics. So we do think that GP75 is a key to the inhibiting of these two proteins. With other data that I'm not showing you, we actually think it's inhibiting both GP63 and GP62. So the last thing I'll show you is that we looked in phages that infect a whole range of different kinds of bacteria. All of these are N4-like phages, and in the purple box is the presence of a GP75-like gene, and in blue and green are the presence of the other holin genes. You can see that they coincide with each other. Our very first experiment, our first two experiments looking for activity in the other phages has not yet yielded any. So we are interested in pursuing whether or not the presence of GP75 correlates with the ability to do lysis inhibition and increase that phage yield by 10-fold or more. So in summary, what I have shown you here is that N4 encodes a complete lysis cassette that can do rapid lysis. And also it encodes a factor called GP75 that we think is inhibiting rapid lysis, allowing for lysis inhibition, accumulation of a large number of phage before allowing for lysis. And this lysis inhibition pathway is of interest to us because if the phage choose to stay, we get a larger yield, and that is something we'd like to be able to control in many different bacteriophages. So to bring this all back out, I've told you about how bacteriophages are potential alternatives for clinical treatment of antibiotic-resistant bacteria, and how we focus on finding effective, safe, and useful phage for killing bacteria and potentially using their proteins on their own. And we also are studying how to improve the production by taking advantage of pathways that phage already use for increasing their own yield. With that, I will thank all the individuals who are currently in my lab listed here. So the two primary projects I told you about are Michael and Michael's, and I would like to thank the funding from my department and university, as well as the National Institutes of Health, as well as my hosts. Thank you for your attention, and I'm happy to take any questions. Gracias.
Muchas gracias, doctora, por su seminario. Quedamos abiertos a sus preguntas.
Thank you very much for your talk. Phages are always fascinating, and your research really is very illustrative of this fact. I want to ask you something that you didn't talk much about, which has to deal with the production of phages for therapeutic uses. I wonder if you already have some experience, because I think it will involve isolating the pathogenic bacteria, then selecting the phages that are infective, and then you will have to grow these very dangerous bacteria in a large scale, I assume, to obtain the phages. Has this been done, and what are any concerns about this approach?
Yeah, that's correct. The phage are usually very specific for the host, which means that you do have to grow the pathogen in the lab, and that's why this is not a simple task. Sometimes you can know the receptor and move it into a lab strain of E. coli, or you can either knock out or remove some of those pathogenicity factors that allow it to be safer to grow at larger scales in the lab, but not always. And that is one of the challenges. There is an effort to generate banks of related bacteria that could potentially be used as alternative production hosts in these cases. There are also efforts that are directed at in vitro reconstitution of phage that would allow you to be able to make them and make changes in them without having to grow large amounts. But once you've grown those large amounts, you do still need to purify them away from the debris of the bacteria. And particularly for gram-negative bacteria, that involves the removal of LPS. And for gram-positives, there are also molecules that, if not removed during that purification process, can on their own induce fever and other kinds of detrimental immune responses in the patients in the clinic. Therefore, there are certainly challenges associated with the production of phage for any kind of case. And those have to be met, unfortunately, on an individual basis, depending on the pathogen of interest.
Thank you.
Thank you for your question.
¿Alguien más? También podemos recibir preguntas desde nuestro canal de YouTube. Si alguien nos está viendo por ahí y quiere escribir una pregunta. ¿No? Sí, seguro.
Well, I have another question. Are there, you presented one example of one person that was saved by this approach. Are there any other examples, or there is only one
There are many
example?
other examples. Yeah. So that particular case that I presented was the first widely publicized case in the United States in 2016. However, since then, there have been many additional case reports in here in the US. But before that, I would like to emphasize that since 1920s, there have been phages being produced in former Soviet Union countries, Georgia, Poland, Russia, and Belgium, Czech Republic, all of these places have been treating people with phage almost continuously since then. Now, at large scale, that's different. And today, everywhere, on most of the populated continents, all countries are working on how to produce. And there are individual physicians who, if they are interested in pursuing this, will be sought out by patients that can work with the clinics that can produce and treat these people. So there are people who have had all different kinds of infections, respiratory infections, heart infections, muscle infections, urinary tract infections, wound infections, all kinds. They're mostly individual case reports. What we don't have very many of are large-scale clinical trials that are experimentally controlled.
Okay, thank you.
Yep.
Gracias. Si alguien quiere hacer preguntas en español, la doctora habla un perfecto español, entonces con toda
Sí, con gusto
confianza.
escucho preguntas en español, no hay problema.
Yo tengo una pregunta, doctora. En estos países donde uno puede ir a la farmacia y sin necesidad de una receta, adquirir una terapia de fagos, ¿cómo podría esto incrementar el riesgo de una posible adaptación de las bacterias hacia esos fagos?
Es buena pregunta. En esos casos no hay ningún médico que está siguiendo a ese paciente, entonces es muy probable que en esos casos van a desarrollar esa resistencia bien pronto. Y una cosa que a mí me da pausa en esos casos es que ese paciente ni sabe si esos fagos van a poder infectar las bacterias que le están causando problemas. Entonces, ese método de simplemente tenerlos disponibles para cualquier que quiere venir a comprarlos no me hace la mejor manera de abordar este problema. Yo digo que es mucho mejor usarlo como una medicina personalizada o en un hospital donde se sabe el 80% de estos pacientes se infectan con este tipo de bacterias. Así tenemos la mayor probabilidad de tener la esperada, lo que uno espera de un tratamiento así, en vez de tener el mismo problema que tenemos con los antibióticos, es primeramente eliminar esa bacteria o curar a esa persona para que ya no después tengan que repetir ese tipo de terapia.
Muchas gracias. Sí.
Gracias por la presentación. Yo solo una pregunta sobre los mecanismos para liberar los fagos. No sé, en un momento ya hablaba sobre la especificidad, pero mi pregunta es si no hay alguna reacción contra, no sé, algunas bacterias que sean benéficas o que habitan en la microbiota humana que puedan afectar, o aquellos que se aplican en la piel. O sea, supongo que varía si es un vehículo líquido, sólido, o acerca de eso también hay, supongo, bastante que hace más complejo, ¿no?
Sí, es buena pregunta, gracias. En caso de los seres humanos, no hay muchos estudios con respecto a cambios en el microbioma cuando hay un tratamiento con fagos. En los ratones y a veces en diferentes modelos sí hay ese tipo de información. Pero normalmente lo que decimos es que no va a haber ningún efecto en otras bacterias, pero la realidad es que eso no es cierto. Hay varios estudios que están enseñando que cuando un fago pensamos que solamente infectaba a un tipo de bacteria, puede infectar a otros. Entonces hay probabilidad de un poco de cambio, pero no es un gran riesgo porque tenemos tantas diferentes bacterias en nuestro microbioma. Cuando se trata de la piel o cualquier lugar en el cuerpo, no es solamente una bacteria, ni si fuera una de las que es más abundante, sería tanta problema para el cuerpo adaptar a ese cambio. Lo que nosotros más tenemos en cuenta es que no vamos a introducir un fago que ayuda a mover los genes de resistencia o algún factor de virulencia. Eso es lo que nos preocupa más.
Ok, gracias.
Una última pregunta, Jolene, simplemente para aprovechar tu expertise. Hace poco se publicó un artículo respecto al diseño inteligente de fagos de manera artificial, ¿no? Y ha dado un par de tumbos aquí en nuestro instituto, en algunos correos, con algunas opiniones, y me gustaría realmente escuchar la opinión de una experta.
Ese artículo cuando salió el año pasado, ¿verdad? Antes que
Sí.
lo habían. Es bien reciente que se publicó, pero salió en preprint el año pasado. Y lo que hicieron fueron entrenar una inteligencia artificial sobre todo lo que hay en los virus, el mundo de los virus. Y lo que salió cuando querían algo completamente nuevo fue algo que era bien similar al fago phages 174. Y en mi opinión, lo que está haciendo la inteligencia artificial es simplemente cambiar lo que ya está ahí. No es algo tan nuevo porque hacer tantos cambios y hacer algo nuevo no lo ha hecho. Es muy similar a lo que ya tenemos con respecto no simplemente a la secuencia, pero a lo que salió, las proteínas y la estructura de ese fago. Entonces, para la inteligencia artificial crear algo nuevo, en lo que yo he visto en ese estudio y en otros lugares donde están intentando eso, todavía no hemos llegado a ningún nivel para que eso pase. Sí, no sé si hay otra opinión de que querían discutir o un aspecto en particular, pero eso es lo que yo pienso.
Creo que has dado una muy buena respuesta. Muchísimas gracias. Una pregunta más, perdón.
Hola, muchas gracias por la presentación. Quería preguntar si hay alguna perspectiva del uso de los fagos para contender o atacar la microbiota de los tumores. Vaya, en el caso
Mm,
de pacientes que no puedan tomar antibióticos o en
ujú.
algún caso que se pueda atacar esa microbiota bacteriana que hay en los tumores que luego evita que los medicamentos antitumorales funcionen.
Mm. Eh, Gracias por la pregunta. No estoy muy al tanto con respecto a ese tipo de problema con microbiota en los tumores. Lo que sí he visto es que están intentando usar células infectadas con fagos, bacterias infectadas con fagos, inyectándolos en tumores para atraer al sistema inmune para que ataque. No sé específicamente sobre poder matar a cualquier bacteria que que es un problema con el tratamiento. Dices que el tratamiento no funciona porque la bacteria está o comiendo o algo cambiando lo que están usando para ese tratamiento, ¿verdad?
Sí, dice que sí.
Entonces sí, si se conoce cuáles son esas bacterias, entonces sería posible porque resulta que los fagos son muy buenos para mover entre los tejidos del cuerpo. Entonces sí sería posible si se conoce cuáles son las bacterias.
Sí, bueno, pues muchísimas gracias a nuestra invitada del día de hoy y muchas gracias a todos los asistentes. Gracias, Jolene. Seguimos en contacto.